of gene editing. We'll start with the basics. You can pause through to see the slides and read them if you want to, but this is standard 7th grade science curriculum um, on genetics and heredity. After that, we will learn the 7 scientific reasons on why gene editing is dangerous and unpredictable. Next are the benefits with the cons, and at the end you may ask me any resources or for questions. You will see the sources and you may ask me any questions for clarity. The basics. And then this is how gene editing actually works. So you start with agrobacterium, then there are targeted DNA segment that is removed and a new DNA fragment is placed. From there on, there's culturing modified agrobacterium. So the modified bacterium is injected into the plant and the plant is grown and sent into our food supply. Now, there are 7 reasons why gene editing is dangerous and unpredictable. Biotech companies falsely claim gene editing's precision, pressuring governments to eliminate safety tests despite at least 7 reasons why it's false. CRISPR's genetic scissors guided to specific DNA locations often cause off-target cuts due to the genome's similar sequences. From there on, there's sloppy repair. Cell repair mechanisms activated by genetic scissors can lead to unintended DNA alteration, including a phenomenon linked to cancer called chromothripsis. Mixing genes. Contaminating DNA fragments can be permanently inserted during the reconnection process, leading to unpredictable mutations in gene-edited organisms. Proteins. Gene editing can fail to disable genes 1/3 of the time, resulting in potentially toxic or allergenic mutant proteins in genetically modified organisms. Insertion of gene editing scissors into plant cells or instructing cells to produce them can cause hazardous mutations, including increased allergens or toxic, which is insertion damage. Mutations. Cloning gene-edited organisms in labs can cause extensive genome damage, resulting in hundreds or thousands of mutations. Unpredictable epigenetic changes in gene-edited mice persisted through 10 generations, with significant chromosomal damage reported in Nature, raising concerns about heritable genome editing's safety due to assessment limitations. Here are the so-called benefits of gene editing. There are some benefits of gene editing, removing toxins, and solving problems if it's beneficial, right? But gene editing can add certain mutations and more to your food instead of making it healthy too. It just depends on the food and the exceptions. Gene editing may solve world hunger, but it can also cause people to die due to cancer and the damage it does to our food and us. Gene editing speeds up the process of evolution, but it also ruins the natural selection process. Lastly, gene editing may seem to be predictable and safe, but it is actually quite the opposite. Here are all the sources I went through. You can look through these for more info. And if you have any extra questions you may ask me them, I'd gladly answer. And thank you for your time. Have a great day.